Good evening, Carmarthenshire. Imaging atoms, molecules and cells. So this evening we're going to be looking at imaging and how we interpret images and how we analyse images. And I'll be showing some examples from various projects that I've been working on over the years. So we'll start with the world of atoms, because I started imaging, at least professionally, when I was looking at atoms and molecules on surfaces. I'll be then taking a little bit of an aside because I want to make sure people realise the difference between what's actually out there and what we think we see when we look either at the real world or at images of the real world. And that explains why sometimes interpreting images and analysing images can be such a challenge. I'll be looking at spin-offs of some of the work I was doing in those early years, and the spin-offs include looking at applications in earth sciences and applications in medical sciences. And given that I'm currently working in the area of medical science, I'll spend a few slides looking at those in more detail, imaging relatively complex systems and finishing with a look at how we use imaging and other techniques in investigations of cancer. So, in order to make sure that we're all on the same page, as it were, because we're talking about images and how we try and extract information from images, there are two important concepts. One is the idea of image processing. And image processing means taking an image and doing something to it to help us in the interpretation of what it is we're looking at, an interpretation of the information in the image. Image analysis means taking an image and doing something to it, not necessarily to change the image, but to extract some quantitative information from the image, generally extracting some numbers which are useful to us, quantification. So image processing means how do we interpret what we're seeing. Image analysis is how do we get some numbers out of what we're doing. And as I said earlier, in this particular talk, I'll be illustrating what I mean with research projects from, in some cases, many years ago, and towards the end of the talk, something more recent. It's perhaps surprising to realise just how far we can go in terms of imaging the very, very, very small. So here's an image, not one I've taken myself, but here's an image taken with a microscope that's able to obtain images at very high magnification. The scale of this image on one side is 850 picometers. Remember, a picometer is 1,000th of a nanometer. So this image is less than 1 nanometer across. And 1 nanometer is 1,000th of a micron, and 1 micron is 1,000th of a millimeter. So if we imagine if this is the magnification we're looking at, in this case, atoms of carbon on a surface, we can ask ourselves, well, if we use this magnification, well, what would the size of a grain of sand be? And the answer is a grain of sand at this magnification would be about the size of the moon. So whenever I see images like this that show individual atoms or individual molecules, I do tend to think of the William Blake poem, "To see a world in a grain of sand." The technology that allows us to do that is called a scanning tunneling microscope, and that's what's in the background behind the rather handsome scientist there in the foreground. That equipment in the background houses a device called a scanning tunneling microscope, and that's a microscope that allows us to see at these incredible magnifications. I'm not going to go into too much of the details, but it's not a visual technique, it's not an optical technique. It involves taking a very sharp needle tip and moving it across a surface very close, not quite in contact with the surface, separated by only perhaps nanometers from the surface, and through a particular phenomena which is quantum mechanical in origin, and I'm not going to describe in any detail, but through the wonders of quantum mechanics it allows us to tell where that tip is relative to the surface, and it allows us to map out the structure of a surface. So for instance, this image taken by a colleague of mine in the University of Manchester, this shows the surface of rust. So the surface of rust is made up of iron atoms and oxygen atoms, and it just so happens that this particular surface is made of two phases of iron. For those that remember their chemistry, there's Fe2O3 and Fe3O4, and we're looking at individual atoms on the surface here. Each of these tiny bumps is an individual atom on the surface. Note the scale here, there's a 2 nanometer scale bar. So these microscopes, these rather incredible scanning tunneling microscopes, are able to show individual atoms and molecules. And they've really revolutionized over the last 20 or 30 years what we've been able to see and what we've been able to understand in terms of how atoms interact with each other. Working with scanning tunneling microscope, or STM, images led to me to make some software that I thought would be useful for my students. In other words, my PhD students were analysing images such as the one we've just seen, and in order to make it easier for them to process and analyse images from these microscopes, I wrote some software to allow them to do that. But there are lots of scanning microscopes. Some of them you won't have heard of. You might not have heard of a scanning Auger microscope. You possibly have heard of a scanning electron microscope, which again gives you very high magnification of very small areas of a surface. And there are lots of other scanning microscopes: scanning force microscope, scanning ion microscope, scanning laser microscope, scanning optical microscope, and we've just already seen scanning tunneling microscopes. So there's an awful lot of microscopes that fall into this category of scanning something microscope. And that's why I ended up with a piece of software that I thought would be useful for all such microscopes, and I used the term SXM to mean a scanning microscope where the X can mean almost anything. And that led to the unpronounceable software of Image SXM to show that it's imaging software that can be used for lots of scanning microscopies. I started writing this software more than 30 years ago. And it's had 50,000 downloads in the last decade or two. And it's used by researchers at universities and research centres around the world. And here's a map with a pin in each country that I believe has some users. You can see that I'm not doing so well in Greenland, and I haven't yet broken into Antarctica. But apart from that, the software is being used around the world, which is very satisfying to know that software that I wrote for my students ends up being useful to other people. I used Image SXM and various microscopes to look at how atoms are put together. So this is if you like, a set of models of how we think atoms are interacting with each other, how they're arranged, the geometry of atoms in various crystal structures. Most metals and a lot of semiconductors have crystalline structure, and if we look at how the atoms are arranged in those crystals, we get a lot of information about how those atoms behave, how they're interacting with each other, how they will interact if other molecules are brought close to one of these surfaces of these crystals. This is a scanning tunneling microscope image of a particular surface of a metal called scandium, and I worked on that metal and many other in the family called rare earth metals. I studied these metals for quite a long time using a combination of the sort of microscopy we've already been looking at, but I also used spectroscopy using light of various wavelengths, typically ultraviolet light, and also some diffraction techniques. Diffraction means firing particles at a surface and seeing how they behave when they come back off the surface, and the patterns they make tell us about the arrangement of atoms on the surface. And I spent a couple of decades looking at rare earth metals and ended up writing a book there at the bottom, The Structure of Rare Earth Metal Surfaces. I wrote that book with my postdoctoral research fellow at the time. Let me show you the sort of thing we can do with a little bit of image processing and image analysis and a little bit of imagination as well. Here's an image taken by a colleague of mine. He doesn't like being named simply because it's not a very good image. It was an image taken a long time ago, and since then he has got a lot better at imaging these surfaces. It's the surface of a particular alloy that I'm, again, not going to describe in detail. But I like this image precisely because it is not very clear, and it makes the point that you can extract a lot of information even from images that are not particularly crisp and clear. What we can do with an image like this is to say, well, we expect that the atoms are arranged in particular patterns. Can we use that information to pull out a more clear image of what's actually going on this surface? The large white blobs, by the way, are contamination and are not what we're interested in. We're interested in how are all the other blobs in the background, the mottling that you can see here, how are all these atoms arranged? And we can apply certain processing techniques that effectively pull out the patterns of how the atoms are arranged. And again, I'm not going to go into detail because it's quite mathematical, but we can use a particular technique, a particular process that effectively pulls out the patterns of where the atoms are placed. And it may not be immediately obvious, but the pattern of dots on the right-hand side actually follow a 10-fold symmetry. In other words, the pattern of dots looks pretty much the same if you were to rotate them by 1/10 of a revolution or 2/10 of a revolution. And using that information about the pattern, we can reconstruct what the surface would look like if there wasn't any of this contamination or anything else getting in the way. And that's what we would get. So this, we think, is what the surface would look like if we had no contamination and if all of the information in that image was made as clear as possible. And you might say to yourself, well, we've processed the image, but are we sure we're not kidding ourselves? Are we sure we're not actually doing something to that image and imposing something on the image that wasn't there in the first place? Well, that's a very valid question, and we can do a very quick double check. For instance, we can look at this feature here shown in the blue circle. That looks like a 10-fold sunflower, if you like, a 10-petaled sunflower. So is that real, or is that just an artifact of the fact that we're looking for things that appear to be 10-fold? Well, if we look very carefully at exactly the same position in the original image, and there it is shown in the red circle, well, there is a 10-fold pattern in there. There is a 10-fold sunflower of atoms. We have simply made it more clear by doing this processing. It was there in the first place. We've brought out those features and made it clearer and more contrasty, made it easier for us using our eyes and our brains to actually look at that and say, yes, I can see how those atoms are arranged. And that's what I spent a fair bit of my time doing, looking at images, saying, what can we do to that image to make the information, to make the content more readily accessible to humans to see what's going on? In this particular case, a colleague had produced a surface covered with atoms, these little triangular things that you can see. Again, ignore the very bright blobs. They are just some level of contamination. We're interested in these little triangles on the surface. And those triangles can be in one of 4 orientations shown in the panel on the left-hand side, and represented by the icons on the left, blue or red triangles pointing up or down. And so I wrote software that would look at these surfaces and extract for each of those molecules, which way up are they, and is there any particular pattern in the way these molecules are sitting on the surface? It was not clear before we did the analysis whether or not these molecules would prefer to be the blue way up or the red way up or pointing down. But once I had done this analysis and extracted that information, then the image on the right hand side is a representation of those molecular orientations, and now it's easier for humans to look at that and say, is there a particular pattern? Are there more blues than reds? Are there more triangles pointing up than pointing down? It turns out that there was quite a mix of both, but we didn't know that until we actually did the image analysis. This is the sort of image you've seen before, especially for me. If we're looking at not atoms on surfaces, but large scale, for instance, looking out into the universe, it might be that the images we take really don't need much processing. So here, under the very dark skies of Africa, is an image of the Milky Way I took a few years ago. And because there's very little, as it were, contamination, very little light pollution stopping us from imaging what we're interested in, the Milky Way and the stars and the planets, we didn't need to do much to the image. But if we were to take a similar sort of image but from the UK, especially if we do it from a city, then we'll find that because of the light pollution that we tend to find in towns and cities, it can be a lot more difficult and require a lot more image processing to extract the information that's in the image. So here, for instance, is an image I took of a nebula, a big cloud of hydrogen, which you can probably not see at all. There's a bright star over to the left and a load of stars in the background, but you can't see any of the image of the detail that I was looking for when I took this image. However, if we process the image, which often involves taking lots of images and adding them all together to get, in effect, a much longer exposure, then we can start to see some of the information that's coming out. And I used ImageSXM, although ImageSXM was the software that I wrote for scanning microscopy, looking at atoms and molecules. It so happens that that same software could also be used for analysing images of the sky. And so I wrote a lot of the routines for astrophotography into ImageSXM, and I used that for many, many years. And that middle image, which now starts to show the nebula that I was interested in, it's actually called the North America Nebula, and you can now get the hint of why it's called that, because it has an outline that's sort of reminiscent of the North American continent. And with that image, you can then tweak it a little bit and bring out the colours that are embedded there. So remember, again, we're processing in the sense that all the information shown in this right-hand side, all of that information was in the original images, but it's been processed in such a way that the detail that we're interested in is brought out and made more readily visible to us mere humans. In a sense, the computer already knew it was there, but humans need a bit of extra help so that we can see what's there. So that brings me on to this idea of the difference between perception and reality. In other words, what we see and what's actually there. And I'm taking this particular set of examples to make the point that processing images and image analysis can be a real challenge, because what we think we're seeing is not necessarily what's actually in front of us. How we perceive images, in other words, what we think we see, can be very different from the actual information content. Now, in most day-to-day situations, we don't worry about that. We just trust our eyes and say, well, if I can see something in front of me, then it's clearly there. However, we do have to worry ultimately about the difference between what humans see and the difference between what computers see. We can ask, which does a better job at image processing? Is it a human brain or is it a computer? But bear in mind that they're both computers. A brain just happens to be a computer made of carbon, and what we normally call a computer is a computer made of bits of silicon. So we could ask, well, which is the better image processor? Stuff made of carbon or stuff made of silicon? And the answer is, well, it depends. So let me make this point to hopefully an audience that doesn't have any colour blindness. So I'm assuming that you can all perceive colours, and I'm guessing we all perceive colours the same way. And I'm hoping that the screen you're currently looking at, whether it's a computer or an iPad or whatever, shows colours reasonably good fidelity. So I'm not going to ask you to say anything, but I'm going to give you a few seconds to think about the answer to this question. If we have a look at this cube on the left-hand side, a sort of big Rubik's cube, I'm interested in the colour of the tiles on the top face. Perhaps you can see that there are 5 by 5, so there are 25 tiles there. I'm asking you, and I'll give you a few seconds, how many of those tiles are blue? Okay, make a mental note of that answer, and then have a look at the cube on the right hand side of this graphic. Again, 5 by 5 tiles on the top face of this cube. Of those 25 tiles, how many of them are yellow? So I hope you now have let me answer that question. I hope you now have 2 numbers in your head. And if you have normal colour vision, you will probably come to the conclusion that there are 4 blue tiles on the left, and you'll probably come to the conclusion that there are 7 yellow tiles on the right. However, the point I'm making in this particular graphic is that you're wrong. The blue squares on the left and the yellow squares on the right are actually the same colour. Yes, I'll say that again. The 4 blue tiles that you identified on the left are actually the same colour as the 7 yellow tiles that you identified on the right. And at this point, you probably don't believe me. So what I'm going to do is take away all of the pixels other than those 4 tiles on the left and the 7 tiles on the right, and we'll see what they actually are. So let me take away all the other pixels. That is what you are actually looking at. When you thought that they were the blue tiles on the left and the yellow tiles on the right, they're actually all grey. And they're actually exactly the same shade of grey. Let me put all of the confusing pixels back around them. I guarantee you that the original pixels have not changed. All of the surrounding pixels have changed, but those things that you think are blue are actually grey. Those things that you think are yellow are actually grey. They only look blue because they're bluer than all of the yellow pixels in the background. And these yellow ones that you think are yellow only think are yellow because they're yellower than all the blue pixels in the background. The colour vision of humans and a lot of other animals works on the basis of contrast. You do not see absolute colours. You only see, in effect, colour differences. So you only think they're blue and you only think they're yellow because of surrounding pixels. You cannot, with your human colour vision, tell exactly what a colour is. So whenever you look at anything, whether it be something in front of you or whether it be something on a computer screen, bear in mind that colour is perhaps not what you think it is. Sometimes your colour vision is fine, but it can easily be fooled. And if you don't believe me, you can go to the webpage on the left-hand side, lottolab.org, to convince yourself that I'm not hoodwinking you. And I'm trying to convince you that perception is not the same as reality, and it's not simply a question of colour. Let me just take those away again and jump to the next one. You're not even very good at judging one thing being brighter than another. It's not a question only of colour. It can be simply which one is brighter. or which one is a darker shade of grey. So for instance, in this case, we have two squares on a checkerboard, one labelled A and the other labelled B. It's the square we're interested in, not the shade of the label itself. So is square A darker or lighter than square B? Most people will come to a conclusion. It doesn't take you long to come to an answer. But let me show you what happens if we block out pixels to the left and we block out pixels to the right. Now, when we look at the square labelled A, it's exactly the same shade as the rectangle on the right or the left. And it's exactly the same for square B. It's exactly the same shade of grey as the rectangle to the left or the rectangle to the right. So the square labelled B is exactly the same shade, not similar, exactly the same. Now that you know that square A and square B are exactly the same shade of grey, let me take those two masks away. I haven't changed A and B, but what is your brain now telling you? What is your eye and brain telling you about the shades of A and B? Even though you know they're the same, because I've just shown you they're the same, it is almost impossible for your brain to come to the conclusion that they are the same. Your brain is so easily fooled. Computers don't suffer the same confusion. If you want to know how bright a pixel is, the computer will tell you. It's not fooled by the context of the checkerboard or this green cylinder, which appears to be casting a shadow. So why is that important? Well, it's important because when we look at an image, remember, what we see is not necessarily what's there. So if we want to analyze this image, for instance, here's an image given to me and somebody said, I've grown or I've deposited a whole load of strands of DNA. So each of these little white worms on the surface is actually a strand of DNA, and it's on a glass surface. You might say, why is the background all mottled? Because if it's on a polished glass surface, that should be flat. Well, it is flat on the scale of, let's say, a micron or so, something of order the wavelength of light. But if we look at it with the resolution of nanometers, we find that even a polished glass surface still looks quite rough. So you say, okay, if you wanted to know how many strands of DNA have I got there, you look at that and you would say, well, no problem, I can count them, 1, 2, 3, 4, 5, 6, and a human can easily look at that and say, well, clearly the DNA strands are brighter than everything else. And in this particular image, bright means high and dark means low. So we're looking at effectively the topography of a surface, and all of the DNA is high and therefore looking quite bright. And if you say, well, I want the computer to count these for me, you might say, well, that should be easy. I can see what's going on, so the computer ought to be able to see it as well. No, perception and reality are quite different things. So you think what you're looking at is a little bit of dark background with a little bit of roughness in it, and every once in a while there's something quite bright. So in other words, if we were to ask what are the intensity variations, or the height variations if you like, along a particular line, you think, well, I've got a background of glass and then every once in a while I see a strand of DNA. That's actually not what you're looking at. What you're actually looking at is something that looks a lot more like that. It is a lot more messy than your eyes are telling you, or your eyes and brain are telling you. So it's actually the profile of how the intensity varies along that red line. So if we ask ourselves which of those peaks that we can see there correspond to a piece of DNA, it's almost impossible for a computer to pick it out. If we look on the left-hand side, we find that that red line crosses a piece of DNA 3 times, as indicated by the orange arrows. How does that correspond to this on the right? Well, it corresponds to that, that, and that. How on earth earth is the computer supposed to work out that those three are the peaks corresponding to DNA, and all the other peaks in this profile are simply the background mottling of glass? We look at the image and we can figure it out. The computer, well, that needs a lot more help, if you like, to figure out that those three are DNA and the other lot aren't. So analysing this proved much more difficult than you might guess simply by looking at it, because humans are very good for picking out patterns. Computers have to be taught how to do it. So in this particular case, I had to take the original image on the left and process it in a particular way using an algorithm that I'm not going to describe, and process it in order to produce this black and white version where the DNA is absolutely unequivocally clear against a background which is pure white. In other words, I had to turn the rather mottled image on the left to a black and white image such that there was no doubt it was absolutely black and white, metaphorically as well as literally, so that there was then no confusion when the computer was asked, well, how many strands of DNA are there actually there? So in other words, things that look easy can be very difficult for a computer to do, and sometimes vice versa. Some things that look horrendously complicated are actually quite easy for a computer to deal with. Human brains and computers are very, very different things. Let's have a look at some of the spin-offs of what I've been doing over the years. Applications beyond physics and chemistry, which is effectively where I started, are a natural spin-off of writing software that can do a lot of different things. So for instance, there were spin-offs in earth sciences where the software was used to look at essentially rock samples, and there were spin-offs in the medical sciences, and I've done a lot of work on applying software to medical science or medical physics areas, which I called software for medical applications. Again, I'm not going to go into horrendous detail because it gets a bit mathematical, but if you were to take a piece of rock and produce a very thin section, it would look like the top left. In other words, very thin sections of rock are almost transparent. But there's a lot of information in there which we can't readily see, information about how the atoms are arranged in crystals that are oriented at particular angles. And we can start to pick out information if we pass the light through polarizing filters in the microscope. That allows us to pick up information that relates to the polarization of light and its relationship to the angle of the crystals in each of these little crystallites of rock. And as I say, I'm not going to go into detail, but I wrote software that allowed me to take lots of images of rock samples taken through different polarizers at different angles, and they were processed and quantified, and I worked out where the boundaries were between these crystallites, and then I ended up color coding an image where the color is not the true color of the piece of rock that we're looking at, but is a color coding of in which direction, here indicated by a sort of compass if you like, in which direction were the atoms arranged in this particular crystallite. And looking at those colors tells us about how the crystallites are arranged, and a geologist can then use that information to glean something about the processes that were involved in forming that particular rock structure. If those of you in the audience that are genuinely interested in how image analysis is applied in the earth sciences, well, I can recommend a very good book written by Heilbronner and Barrett, and it's available at a very reasonable cost if you let me know. But let's have a look at medical spin-offs. Over the years, I've looked at a lot of different applications. I have a number of colleagues here in the University of Liverpool and in university hospitals and research centres elsewhere in the UK and indeed across the world. So I've ended up dabbling in writing code, writing software to help understand particular problems in medical sciences, such as looking at carbon particulates for those that have inhaled carbon or smoke into their lungs, looking at parasite development, looking at microcirculation, circulation in the microvessels in the blood. I'll be coming back to that one in just a moment. Looking at structures on the back of the eye on retinal scans, looking at the morphology of parasites, looking at how cells behave, and looking at bacteria. So I've had a lot of interesting projects, some of which students were working on, some of which I simply worked on myself. One of the latter ones was looking at human chromosomes, which I thought was an interesting problem. The Women's Hospital in the next to the University of Liverpool would often scan the chromosomes of pregnant women and their fetuses to try and determine if there were any chromosomal abnormalities. And I thought it was an interesting problem. Rather than have an expert sit down and look at images like this, I thought, wouldn't it be interesting if a computer could automatically take images such as this and figure out which chromosome is which? That panel on the right hand side gives an indication of what we would expect from a healthy individual, the final chromosome being X or Y depending on the gender of the baby. I thought it would be very interesting. It turned out to be much more complicated and rather tricky, but it was still fun to actually get my head around the problems of trying to identify which chromosomes are present, which way up are they, are the banding patterns that we can see here matching up to the banding patterns that we would expect in a human chromosome. So that was an interesting project. Let me deal with a couple of projects in a little more detail. One of them is microcirculation analysis, and the other, which I'll finish with, is an investigation of cancer. Microcirculation, that was the result of a collaboration with Alderhay Hospital. It was one of the first trials of the miasma software that we did on some patients in the intensive care unit, because meningitis causes sepsis, i.e. blood poisoning, and they were interested in looking at the effect on the circulation in capillaries, i.e. what they call the microcirculation. And to do this, they had a small portable microscope which was placed underneath the tongue of a patient and produced a video that I'm about to show you. So in other words, it's not putting people under the microscope, it's putting a microscope under the tongue of people, especially children. And you end up with a video that looks like this. Bear in mind that the area we're looking at is rather small. It's only a square millimetre or so. And what I hope you can see is a capillary network, and every once in a while you can see individual blood cells passing through the capillary network. The more you look, the more you can see. Some of these capillaries are quite wide and are showing a lot of motion. Some of them are quite narrow and are showing individual blood cells moving. And the idea is that in sepsis, that blood flow is interrupted or very poor. So the question is, can we use a video like this and quantitatively work out how fast these blood cells are actually moving through the capillary network? If they're not moving very fast, the tissue is not getting oxygenated and the patient has sepsis. If all of the blood vessels are moving through very effectively at high speed, then everything is working well. So the question is, can we quantify what's going on in a video like this? But there's a number of different problems, not least being your eye can pick up the motion, but what's actually going on? If we blow up what's going on inside that red rectangle, it's a little bit indistinct. If we were to blow up even further and ask ourselves, what does a blood cell look like? Well, it's very difficult to tell from a few pixels what is a blood cell and what isn't. And then you have the problem that the blood cell is actually moving through a vessel, through a blood vessel, which is essentially invisible. And the network itself, the blood vessels, are not only invisible, but they're moving. Have you ever tried to get a kid to sit still while you put a microscope under his tongue to get a few seconds of video? Any attempt at quantification is going to be complicated by the fact that there will be motion, not simply the blood cells through the vessel network. The vessel network will appear to be moving itself. and there will be translation and magnification and rotation and distortion. In other words, you can't guarantee that the microscope stays fixed relative to the tongue. Therefore, there could be motion as indicated by the red arrows. But it's also possible that the magnification changes slightly, and so the image might appear to get bigger or smaller. And it's also possible, because you can't hold the microscope perfectly still or expect the patient to keep their tongue still, there might be a rotation there as well, again indicated by the arrows. And there could be a distortion because the tongue is not a solid object, it's a muscle, and that can distort. So if the vessel network is moving all over the place, because you probably have all of those motions in there, how on earth can you possibly hope to actually follow what a blood cell is doing relative to a vessel if you can't see the vessel and the vessel network is suffering from all of this motion? So again, your human eye can pick out the motion and you can say, yeah, I can see those blood cells are moving relative to the network. But to get a computer to do it quantitatively, well, that took a huge amount of effort. Not only have you got all of those motions, you've got the fact that the patient may be on a ventilator, the patient will have a heartbeat, which again makes everything change. So it was real challenge to come up with a computer code, and again, I'm not going to go into the detail because it's rather mathematical, but it did prove to be ultimately possible to figure out how to stabilize the video and hence then see how all of the blood cells are moving. And by taking videos from healthy patients and seeing, well, what sort of speed of flow do we get when we look at the speed through all of the vessels we can see in a video? Well, we got, for a control set of patients, it looked like, on the whole, for a healthy individual, most of the blood seemed to be flowing at the rate of 500 microns per second, or half a millimeter per second. Obviously, there's a little bit of spread there, but in general, it's bunched around that particular point. Whereas if we look and analyze videos from patients with sepsis, notice that although in some cases there's still some blood flow, there's an awful lot at the lower end of the speed of blood cell motion through the capillaries. And the further over to the left this distribution is, the more severe the sepsis. So I figured out a way of analysing these videos such that rather than having an expert say this person is healthy and this person has sepsis, it would be possible in principle to take a video and then have the computer analyse and then highlight this seems to be a problem, get an expert in to have a look at this patient because they seem to have abnormal blood flow. I thought the easiest way of conveying this information is not to show histograms of flow speed and the number of vessels which have that flow speed. I thought it was more useful to color code the map of the network to show in a control patient you would expect the flow to be good, in which case the color coding here would be relatively high and would be green, as indicated by the fact that the blood flow appears to be many hundreds of microns per second, whereas in a patient with sepsis you would get a lot of vessels in which the blood flow is rather poor, down here at 0 to 200 microns per second. And so even if you didn't look at the scale, you could glance at a map like this and say, if it's mainly green, the patient is okay. If it's mainly red, the patient has a problem. We need to get a clinician to have a closer look at this patient and perhaps change their medication. So I wrote this in collaboration with medics at Alderhay to try and write software which could be useful for automated checking of patients with sepsis, in particular children with meningitis. That was a collaboration with the NHS. Let's have a look at the final section, which is perhaps the most recent stuff that I've been working on, which is looking at microscopy and a particular type of microscopy called spectromicroscopy. I'll explain that in just a second. Not using visible light, but using infrared light. So it's describing work being carried out through various funding agencies, including Cancer Research UK and National Institute of Health and Care Research. So those grants have paid for what I've been doing for quite a few years now. The intention was to answer the question, can we identify an infrared absorption signature for tissue that is likely to become cancerous? In other words, if we shine infrared on tissue, is it possible that it absorbs some infrared wavelengths differently if the tissue is healthy than if the tissue is likely to become cancerous in the future? So to simplify that question, we're really asking, can we detect cancer before it's cancer? In other words, not diagnose necessarily, because diagnosis often means it's too late. Can we get a prognosis? Can we tell if something is going to develop into cancer? I've been using some imaging, or I should say we, because I'm part of a group at the University of Liverpool Physics Department who are working with clinicians, developing instrumentation, developing software, and developing techniques in collaboration with clinicians to try and answer these questions. So SNOM is an acronym meaning Scanning Near Field Optical Microscope. Let's not worry about why it's called that. It can be used to image, in this case, image cancer cells, as shown by these images here. But it's not the morphology or the shape of the cancer cells that interests us. It's how these cancer cells interact with infrared light. So again, I'm not going to be describing how we generate the infrared light or how we obtain an infrared spectrum. What matters is we can image a sample of tissue such that each pixel is not simply one pixel indicating how bright that particular piece of tissue is. Each pixel is actually an entire spectrum, as shown by this graph at the top right. It's showing us how does this particular piece of tissue interact with infrared light. Does it absorb particular wavelengths, other wavelengths perhaps not? Does it absorb these wavelengths? So it's not necessarily a let's have a look at where the peaks are, because tissue that is healthy and tissue that's likely to become cancerous unfortunately look very similar. So the question, can we use infrared absorption at different wavelengths to identify the tissue type? Well, we have found that yes, we can, but the differences are not large. And that is where the problem lies regarding the analysis. We found that optical spectroscopy, optical imaging, in other words, if you take an image of a piece of tissue using a normal light microscope, you can get some quite nice detail, but you get no information about what's actually happening. What is the chemistry or the biology of what's going on here? Whereas if we use infrared absorption, we can get information about how these cells are behaving. So optical microscopy tells us about how they're arranged, the morphology. Infrared imaging tells us about how they are behaving. And we have found through the wonderful work of one of our students that we can fuse these images together. The infrared doesn't show as much detail because infrared wavelengths are longer than optical wavelengths, but we found that we can fuse the detail from optical and infrared images to obtain what we call a fused image on the right hand side that contains all of the detail of an optical image, but it contains all of the information about how these cells are behaving, the chemistry, the biology, that we obtain from infrared imaging. So what type of tissues are we studying? Well, we have studied esophageal tissues, and we're now studying oral tissues. Epithelium tissue covers all surfaces of the human body. In other words, not just skin, but also the esophagus and the mouth. And after studying esophageal cancer for a while, for the last few years, we've been focusing on a particular condition in the mouth called oral epithelial dysplasia, or OED. This refers to a condition in which there are changes in the cells that make up the lining of the mouth. That could be the tongue, the cheek, the gums, etc. And the question is, we want to improve the accuracy of prognosis so that we can say that if we see something abnormal in the mouth, for instance, there appears to be a small white patch, a small lesion found in the mouth, what we want to know is that a problem? Will it or won't it progress to cancer? Last time you were at the dentist, perhaps the dentist asked you, as well as looking at your teeth, lift your tongue and put your tongue to the left and put your tongue to the right. They're looking for small white patches in your mouth to tell if there's going to be a problem or not. So here's a picture. Hope you're not squeamish. It's nothing too horrible, but here's a picture of a lesion within the mouth. So there's a white patch in this person's mouth on the cheek, and there's a white patch on this person's mouth that seems to be on the underside of the tongue. Now, you can't really tell from looking at it the difference. So a dentist or an individual looking inside your mouth might be able to say, that's a white patch, I think we ought to get that checked out. But just by looking at it, you can't tell if it will transform to cancer or not. And we wanted to know whether or not it would be possible to make a tool to develop a medical device which would effectively examine that particular area of the tongue or the mouth and actually tell what the prognosis was based on the infrared spectrum. In other words, well, which is which? In this particular case, one of them will transform and the other won't, but how can we tell which is which? The infrared spectrum, unfortunately, looks exactly the same. It looks the same, but it's not the same. There are tiny differences in the spectra of lesions that are healthy and will stay healthy, and lesions which are okay at the moment, but are likely to transform to cancer in the future. And what we've been developing in past years is the software that will look for tiny differences in the spectrum taken from different patches of the mouth and determine whether or not from these spectra we can tell the prognosis of what's going to happen. But if the differences are tiny, how are we going to do that? Well, we can't simply sit down and look at these spectra, especially given that when we're looking at lots of different biopsies that might have been taken from people's lesions in the mouth, we might be looking at millions of spectra. And that's where artificial intelligence comes in. Don't be scared of the term AI. What it means is we are writing software which is trying to train itself to recognize the difference between a white patch which is going to stay healthy and a white patch which is going to transform. How do we write the software to tell it how to do that? Well, again, in the next 5 minutes, I haven't got time to talk about all of the details of how we do it, but let me just give you an idea of what training artificial intelligence means. Let's imagine we wanted to tell the difference between 3 types of animal. Let's say we wanted it to tell the difference between horses, octopuses, and dogs. And we want to do that not from the actual animals, but from pictures of the animals. So we want to train a piece of software to tell the difference between horses and octopuses and dogs. What we do is effectively give it as many pictures of horses as we can find, and as many different pictures of octopuses as we can find, and as many different pictures of dogs, different colours, different shapes, different types of dog, different breeds of dog, and we give it lots and lots and lots of data, and eventually it learns to recognise that, these things appear to be horses, these things appear to be octopuses, and these things appear to be dogs. Even though they're all different types of dogs, if we give it enough data to train on, then eventually it learns to recognize the difference. And in principle, that's what we do with, here's a load of biopsies, here's images, infrared spectra from biopsies where we know from historical records that these did not transform into cancer. And here's a whole load of images of biopsies which we know from historical records did transform into cancer. If we get the artificial intelligence software right, then it learns to recognize the difference, and then we apply these algorithms, basically just computer code. We apply these algorithms so that it then recognizes, yep, give me a tissue I haven't seen before and I will tell you whether it will or won't to develop to cancer. But we have to be careful because, as you're probably well aware, artificial intelligence doesn't always get it right. It might get it right a lot of the time if we write the software well, but we always have to check. In other words, in this example, we can train artificial intelligence to recognize horses, octopuses, and dogs. But before we use it, we then test it or validate it by saying, let's go through a few thousand images and make sure it gets it right. We'll give it a picture of an octopus and make sure that it says it's an octopus. And we'll give it a picture of a horse and it will say the answer is a horse and we'll say correct. But every once in a while, we'll give it a picture of a dog and it will say, octopus. No, wrong. That's not an octopus. We can ask, perhaps, why did it get it wrong? Part of the problem with some artificial intelligence is that it's very difficult to figure out why it gives you an answer. But we in the physics department at the University of Liverpool are developing AI algorithms in which we can check why it gets it wrong. In other words, in this particular case, maybe it got confused because the dog was splashing around in water and it doesn't seem to have 4 legs, and maybe it hasn't got the characteristics that it thought it recognized in a dog, and so it incorrectly put this in the wrong category and said it's an octopus. But the important thing is that we train AI routines, AI software, to recognize particular patterns, and then we test and test and test and validate until we are happy that it's getting it right most of the time, and only then will we apply it to an unknown problem and actually verify whether or not it is correctly diagnosing what's happening, for instance, in an oral lesion in the mouth. All this work is going on in what we've called the SciScan group. We're calling that the spectrochemical infrared analysis scanning for cancer. The device we are prototyping is called the Elder Wand, the Liverpool Diagnostic Infrared Wand. Ultimately, in years to come, we would hope to have a medical device which could be used by a surgeon such that if they are, for instance, excising a particular tumour or a particular part of tissue, they could use this wand to verify, is all of the tissue that we're leaving behind healthy? Or is there a danger that any of the tissue we've left behind could become cancerous in the future, in which case let's make sure we remove that as well. So this is what we are ultimately heading towards. We are working on biopsies at the moment, but ultimately we would like to work with a wand that would be in the hands of a surgeon to actually help guide them during surgery. So I've taken you on a little bit of a journey from where I started looking at individual atoms and molecules, trying to make you realize that sometimes getting computers to analyze images is far more difficult than sometimes we think just by looking at images. And there have been many spin-offs to the work I've been doing over the decades in various areas, including earth sciences and medical sciences. And I've given you a taste of what I've done in the previous years and what I'm working on now. Remember, science is never a solo effort, so I must acknowledge all of the students I've worked with, my collaborators, my academic collaborators in physics, people who've worked at Daresbury Laboratory that have provided infrared sources over the years, and the work I've done on developing miasma, the medical applications of the software I've written. I've got various colleagues here in the university and in universities around the world. The bottom right patches show various funding bodies, and I'm grateful for funding over the years from all of these organisations. Thank you very much for listening.